After the successful completion of the 10 chapters, the 11th chapter is being the biotechnology principles and process. That is the 11th chapter in second PU. So it begins with the EFB, European Federation of Biotechnology. So this European Federation of Biotechnology defines what biotechnology means. So the simple meaning of the definition for this biotechnology is the application of technological aspects to the living systems. Application of the technological aspects to the living systems, that is the removing, adding the genes, etc. So after knowing only the DNA is responsible for the protein synthesis, the information required for protein synthesis is stored in DNA and transmitted in the form of code called genetic code. So this is a DNA, that portion of DNA may synthesize many proteins which are very essential for us, which are very useful. After knowing this, when we can cut the DNA and then we can join the same DNA into some vectors which could carry the DNA into the host to synthesize the desirable proteins. So hence biotechnology is such a branch which deals with the application of technological aspects, that is removal of segment, adding a segment, new segment to DNA. So application of such kind of technological aspects to the living systems is biotechnology, and the important branch of the biotechnology is genetic engineering again. So biotechnology adding the removal of the into the living organisms, that is biotechnology, and important branch of biotechnology is the genetic engineering. So genetic engineering deals with the study of the removal of a segment from DNA, adding a new segment to DNA which could synthesize proteins of our choice. This branch of biotechnology is genetic engineering. It involves, once again, removal or addition of DNA segments for the synthesis of proteins of our choice. This is an important branch of biotechnology is genetic engineering. So it dates back to 1972 when Stanley Cohen and Herbert Bayer prepared the recombinant DNA. So once again I will repeat. So the genetic engineering is an important branch of biotechnology which involves the removal of segment DNA, adding a new segment DNA to synthesize proteins of our choice. Such genetically modified DNA called recombinant DNA. The production of recombinant DNA dates back to 1972. The first recombinant DNA produced by Stanley Cohen and Herbert Bayer in 1972 by using the plasmid of Salmonella typhimurium, a bacterium. A plasmid of Salmonella typhimurium, and by removing the gene having antibiotic resistant gene, so we can they added a new gene to the plasmid, and that is the recombinant DNA. Such that is called genetically modified DNA. The chief vector or the carriers are the plasmids. In the plasmid of Salmonella typhimurium, Stanley Cohen and Bayer, Herbert Bayer, they synthesized this kind of recombinant DNA, and they transferred the same recombinant DNA into the similar bacterium. So for a similar bacterium for Salmonella typhimurium is the Escherichia coli. Escherichia coli transmit this. As the bacteria divides this vector, the recombinant DNA possessing the plasmid also replicates. So this is the technique. So for cutting the DNA segment and for adding certain enzymes are required. So restriction enzymes. First that was discovered in the year 1963. So this kind of years studying of all the list, just like in history we have to write all the years also. That is also very important for the NEET examination. Each sentence is very important for NEET examination. So it is in 1963 the first REN produced. The first REN, that is restriction enzyme, produced was HIND2, Haemophilus influenza 2, HIND2. This HIND2 cuts this molecular scissors, restriction enzymes, cut the DNA at a particular point called recognition sequences, which is having 6 base pairs. This is very important, so far not asked in NEET. At 6 base pairs region, a recognition sequence, this HIND2 cuts, or any REN, it cuts at a particular region, especially HIND2 at the region where 6 base pairs are present. And then that can be joined by the ligase. Now it is called the recombinant DNA. Plasmid having recombinant DNA. So about 900 such REN produced from the 230 strains of bacteria. This is also very, very important. And the nomenclature of such RENs. One code word, GSS. So generic name, species name, strain name, GSS, and the number, Roman number, it indicates the order in which the enzyme is isolated. So that is very, very important. Say for example, E-co-re-13. E, Escherichia, genus name, coli, species name, RY is strain name, and the 13 is the order from which it has been isolated. From the strain. That is very important. That is the nomenclature, which is very important for the examination. There are two kinds of the RENs, exonucleases and endonucleases. Exonucleases, it cuts the DNA at the ends, exo outside, exo, and endo within in the middle DNA it cuts. So these are the two types of the RENs, exonucleases and endonucleases. There is also a palindromic nucleotide sequence in the DNA. So say for example, if the such sequence palindromic repeated one, that is say for example GAATTC, if it starts in one strand from 5' end to 3' end. Once again I will repeat, GAATTC, and from 3' end to the 5' end, for this GAATTC, CTTAAG, that is the sequence. So somewhere in the middle, by using REN, such a DNA palindromic nucleotide sequence DNA, somewhere in the middle, just away from the middle portion, cut the DNA can be cut using REN that produces sticky ends. Sticky ends. Later, after inserting the another DNA, this can be stitched by using ligase, biological stitcher, enzyme called ligase. So, how to produce? What are all the steps involved in the production of recombinant DNA? Number one, separation of DNA from chromosomes in the cells. So, separation of DNA from the chromosomes in the cells, it requires a lot of enzymes. That is the separation of DNA from chromosomes. So, within the chromosomes made up of DNA and histones. We know it. And how to remove that DNA from the chromosomes by chemical method, not given in the book, that is by the chemical method. And then how to get into the DNA, that is also very important. And then knowing the sequences which could synthesize proteins of our choice, so that DNA should be cut into the fragments using REN, and placing such the fragments on the agarose gel. Agarose gel can be obtained from the seaweeds like Geranium and Gracilaria, that is the red algae preferably, and then from the seaweeds, agarose gel placing these fragments, that is another step which could, such fragments which could synthesize the proteins of our choice, or irregularly they are placed having different sequences. And then as we know, there is a method called gel electrophoresis. As it is placed on agarose gel, that is gel, and then it has to be subjected to the electricity. And we know it, DNA is negatively charged when the electric field is applied, move towards the positive anode. Smaller the fragments, farther they move. And according to their size, they are sorted out and removed, sorted out. That is called sieving effect. According to size, they are sorted out. And then such fragments are really invisible. They are in the gel only. And then adding ethidium bromide, which is a strain which gives color to such fragments, but still also invisible. Then if UV rays are passed, exposed to UV rays, then they appear like the bright orange colored bands are formed. So bright orange colored are produced, and after this the DNA fragments, by cutting the gel, such fragments can be removed. That is method called elution method. And similarly, after this, finding a suitable vector. Once again I will repeat, the first method is the separation of DNA from chromosomes by the chemical method. Second method, cutting the DNA into the fragments, and then placing the fragments on agarose gel, and then the sieving effector. Next to sieving effector is the ethidium bromide, then UV rays, expose them to the UV rays. Sometimes then I ask you the sequence also, that is very important for your NEET examination. Next method is the elution method, and then finding a suitable vector. Why these vectors are, the plasmids are used as the vectors. Because the plasmids and bacteriophages, they duplicate, they divide independently. That is one reason, and they get well adjusted in the host, that is also important, and they produce large number of copies, and they have more than one recognition sequence sites. More than one recognition sequence sites are present in such the plasmids. These are all the qualities, hence they are the better vectors, that is the plasmids. And then after this, they should be inserted into the cell, using many enzymes. Say for example, so ligninases, cellulases, pectinases are the enzymes which could dissolve the bacterial cell. These fragments has to be inserted to. If it has to be inserted into the plasmid, then we have to first the bacterial cell wall has to be dissolved. So this DNA, not only native, it is also hydrophilic. That is also so far not asked. It is hydrophilic, hence it cannot pass through the cell wall. So by using calcium chloride, the cell wall, this calcium chloride makes pores in the cell wall of bacteria. Then it has to be inserted and frozen first, and then heated to 42 degrees centigrade. Again it has to be frozen. Then only this DNA segment will be taken up by the plasmids. And in animals, it has to be inserted by my method called micro injection, and in the biolistic gun method in case of the plants, and this DNA then it has to be, when it has to be separated, it should be frozen with the cold ethanol. That is also so far not asked, and so that cold precipitates the DNA molecules. After then, we have to differentiate the transfer mate from non-transfer mate. As I told you earlier, the region of ore, where the replication starts, adjacent to this, there are some disease resistant, that is not antibiotic resistant areas are present. Say for example in E. coli, not in E. coli, that is PBR, plasmid of Bolivar Rodriguez, plasmid of PBR, the different parts we can see. Again, this is used as a selectable marker. It consists of different parts. Say for example, say if we start from region of ore, next to region of ore, the PST1, province staris, that is province staris, expansion, not necessary, but province staris, and PUV1, Proteus vulgaris 1. So this is ampicillin resistant region, and next to this the E. coli, and then next to this chloroplast alterna, that is expansion, CLAP1, E. coli 1, and then the next one is HIND3, Haemophilus influenza 3. Next to this, Brahmi 1, and the SAL1, Salmonella 1, and ROP, that is recognition of the protein site which contains PVU2, that is Proteus vulgaris 2. In the last CET it has asked. So this ROP associated with PVU2, Proteus vulgaris 2. So this is about this one. They are used as selectable markers. If, say for example, at the Brahmi one, if we cut and add the one gene, extra gene, this loses the resistance to the tetracycline, and they will not survive on the substrate containing tetracycline, and they survive in the substrate which contains ampicillin. So, whereas the non-transformant, non-recombinant one survives both on ampicillin and the tetracycline substrate. And such substrates can be prepared not only for the ampicillin, tetracycline, can also be for chloromophenical, kanamycin. So for not asked, so these are the antibiotic resistant regions in some other plasmids. But in the PBR, it contains the ampicillin resistant region, that is PST1 and PVU1, and here the Brahmi, Sal, etc. These are all the tetracycline resistant region. And then so the non-transferment, so that is non-recombinant, survives in both ampicillin and tetracycline if any new gene added at the antibiotic resistant region, say for example Brahmi, they lose the resistance to the tetracycline, they survive only in ampicillin. Non-recombinants, non-transferments survive both in ampicillin, thus they can be separated, hence such a PBR used as selectable markers. But preparing two separate substrates for ampicillin and then tetracycline, for kanamycin, for the chloromophenical, it is a cumbersome project. It is a difficult process, hence only one substrate is used called chromogenic substrate. This chromogenic substrate produces color in case of the non-transformate, that is non-recombinant, and doesn't produce color in the recombinant one. Say for example, this is known as insertional inactivation. If any gene is added, the place where the beta-galactoside produces, it doesn't produce beta-galactosidase but produces blue color on the chromogenic substrate. Whereas the recombinant one produces color, but the non-recombinant one doesn't produce color. It is very easy method to separate the recombinants from non-recombinants or transformates from non-transformates. This is another easy method. And the number of such recombinant DNA should be increased by joining them with the TI plasmids, tumor-inducing plasmids of Agrobacterium tumefacium bacteria. And this is facilitated by tDNA, that is for transformation. Transformation will be facilitated by the tDNA. So far not asked. And such plasmids TI, which produces tumor from the bacteria which produces the tumors, they are disarmed. Even retroviruses, disarmed retroviruses are used as vectors. For example, in gene therapy, retroviruses used as vectors. Whereas for Bt cotton and then the Melanogain incognita, that is for the pest resistant, this one in tobacco, plasmid obtained from Acrobacterium tumefaciens is a place. For the synthesis of this insulin, the vector used can be obtained from the plasmid of E. coli. These things are very important. That you will study in detail in the next chapter, that is the 12th chapter, application of biotechnology, whereas the vector used is the number one is Agrobacterium tumefaciens for Bt cotton Melanogain incognita, and plasmid of E. coli for the only one, that is the insulin, and retroviruses for gene therapy that you will study in later detail. And they are can be amplified by a technique called PCR technique, polymerization chain reaction technique. This involves three steps: denaturation, annealing by producing the primers, and then extension. And Tuck polymerase is used. That is the polymerase enzyme which can be obtained from Thermus aquaticus bacteria, Tuck bacteria. Tuck polymerase is obtained, which helps in denaturation and annealing and extension, and then the 1 billion copies can be produced. Finally, they should be placed in bioreactors which contain a culture solution up to 100 to 1000 liters of solution they contain, and they are used to maintain. These bioreactors are used to maintain the temperature, pH, substrate, salts, vitamins. That is the maintenance of this. That is the function of the bioreactor. There are two bioreactors: simple stirred bioreactor and sparged bioreactor. So in case of simple stirred bioreactor, there is the acid-base controller, acid-base pH controller. The different parts are associated with this bioreactor, simple stirred, and stream for the sterilization, and motor foam breaker, and then the flat bladed the impeller and culture bath to sterile air are the parts associated with the simple stirred bioreactor. Whereas the another reactor called sparged bioreactor. Sparged means bubbling. It contains agitator which produces the increased area for the oxygen and gas entertainment. So these are the bioreactors used to preserve or to maintain, as I told you earlier, salt, substrate, pH, whether acid, base, temperature, and all these things will be maintained. The last one in this chapter is DSP, code word, downstreaming, for separation and the purification of the bioreactor, I mean the recombinant DNA. This kind of separation and purification also, finally, it also involves testing the quality of such a recombinant DNAs. That is also very important. The quality control testing, DSP, that means not only the downstreaming involves separation and purification, but also quality control testing. This is about the chapter, 11th chapter. So biotechnology part 1, the principles and process. So read all the sentences in the textbook, and please subscribe, don't forget.